Hello, I'm Daniel Petersheim, and I had the privilege of working with Dr. Piro during the interim of 2021 to work on this project entitled Multimetallic Complexes supported by a PNP NNN-Ferrocene ligand. And I will explain some of these abbreviations and terminologies as we go throughout this presentation. First of all, I'd like to talk about the concept behind this project. On this slide, you see a few small molecules. There's nitric oxide, carbon dioxide, and nitrous oxide. And these are all small molecules that are relatively inert. But in biology, there are enzymes that facilitate chemical reactions with these molecules. They activate them and cause chemistry to occur with them. And so on the left, there's an illustration of an active site of an enzyme that contains a molybdenum atom and a copper atom. And these two metal atoms are associated with the active site where that activation of the small molecule takes place. On the right, there's one that has two iron atoms that are associated together, and that's where the small molecule is activated there. Now, these two metal atoms form a theme with these sites, and there's two metal atoms and they differ either in their identity, as on the left where it's molybdenum and copper, or on the right where they're both iron but yet they differ in their surrounding environment. And so that is a theme that we would like to mimic in synthetic molecules that we will create in the laboratory. That's the inspiration for our project. So what is a ligand? Just to define it, a ligand is an ion or molecule attached to a metal atom. On the top right, you see there's a metal atom in the middle, and then there's some ligands that surround that metal and attach to it. Some are small, some are large, so it could be just one atom or ion that binds to the metal, or it could be a group of atoms, and they can actually be very large. Some bind to the metal atom through multiple bonds, as in the illustration on the bottom, where each of these units attaches to the central metal atom in two different places. And some contain more than one site for a metal atom. In other words, they can bond to one metal atom on this side of the ligand, and over somewhere else they could bind to another metal atom. So our work involves the synthesis of a large ligand. It's still the same, follows the same definition. It's just a molecule that can attach to a metal atom, but ours contains 109 atoms and two different sites for metal atoms to bind, and at each site there are three potential bonds that can form with the metal atom. So it gets quite complex. So here's a quick picture of our ligand, which is quite complex at first glance, but we'll break it down into a few different parts to make it more understandable. I began work on this project in the previous summer and was able to synthesize a very small amount of the new ligand. You can think of our ligand as containing three different parts, and the backbone is ferrocene, this molecule seen here, and you'll notice that there is a metal atom at the center, an iron atom, but that is a structural part of our ligand and does not take place in the small molecule activation that I mentioned earlier. For that, we'll need two sites for two more metal atoms. And to the ferrocene, we add two different arms. The first one we'll call the NNN arm, and that is the added structure that you can see on the left. And you'll notice the nitrogen atoms in blue on the right, which help you identify that. And that is where another metal atom will be able to bond at that site, be able to bond to those three nitrogen atoms. On the other side, there is an arm that we call the PNP arm, where there are two phosphorus atoms in yellow, as you see on the right, and a nitrogen atom in blue, and those provide another site for a metal atom to bind. And notice that those sites are different. They are not equivalent, and so we are hoping to create two different environments for two different metal atoms to bind and hopefully induce some interesting chemistry that can take place in concert between those two sites. Here's just the same illustration from the previous slide, but it has some red atoms where the metals will bind. On the right is another useful byproduct of our synthesis, that we were able to obtain an appreciable amount of this compound as well, this ligand, and it contains two different PNP arms, which do not provide two different unique environments for metals, but it can serve as a useful reference to our products that we form with the non-symmetric ligand. I've been able to make some progress in new areas this interim. First of all, I was able to refine and scale up the synthesis of our ligand, and you'll notice once again that our primary target ligand is on the bottom, and at the top right is the useful symmetric ligand that we also were able to obtain. Another question to be answered was what metals can bind with our ligand? Once we have it, what metals can we react with it? We experimented with copper, zinc, cobalt, nickel, chromium, and palladium. There's an example reaction at the bottom of the slide, but note that we do not know exactly what the final product is. That is one possibility on the right. And we began the characterization of our products, but faced several obstacles, which I'll talk about in a little bit. Characterization, or you can think of it as understanding our products, it seeks to answer some of these questions. Does the metal bind to the ligand? That's the first thing to know, and is probably the easiest question of these to answer. But then there are also some other questions that are sometimes more difficult and present some obstacles to our understanding of the products. Does the metal bind to one site or to both? Does one metal atom bind to two different molecules of the ligand? In other words, a metal atom might bind to the PNP site of one molecule of ligand and the PNP site of another molecule of ligand. So that is another possibility. Does it form dimers, which are two molecules of the metallated ligand that associate with each other? Or do the metal atoms enter or leave the sites relatively easy? That's another possibility that, if that is the case, it makes it difficult to understand the structure and things like that to characterize it. One of the primary tools for characterization is NMR. We use this extensively on the way to creating our ligand, and this is the NMR spectrum of the asymmetric ligand, the FCPNP NNN ligand that you saw earlier. And notice the narrow peaks and how they're spread across this spectrum here. You don't have to understand exactly everything, but to know that those peaks, those distinct peaks, help us to understand the environments of different protons on our ligand and help us understand what we're working with. So what happens when we react this with a metal? Here's the product that I showed earlier of a zinc, the reaction with zinc, and the possible product shown on the slide. And notice all the different peaks. If you can tell the difference from the last picture, this NMR spectrum has many more peaks at the bottom, and though they are distinct, there's many more, and it's difficult to weed through those and understand exactly what's happening. So that creates more difficulty for understanding this. Here is the NMR spectrum for our proposed copper compound, and notice that the peaks are broader, and this suggests to us that the copper ions may not bind strongly at the two different sites of the ligand and maybe moving in and out and causing some broader peaks. NMR stands for nuclear magnetic resonance, and so it's related to the magnetic properties of compound. Here on this spectrum you see very broad peaks that make it very difficult to conclude much from this spectrum about our compound here, and we believe that is because there is magnetic properties of the compound of the nickel in this compound, and it creates spectra like these that are very limited in their effectiveness. Here's a table that shows some of the reactions that were undertaken, showing the reactant, the ligand, and also some of the conclusions that we've been able to draw thus far about the products or the reaction. Some possibilities to explore in the future. A logical next step is to use X-ray crystallography, and this is a powerful tool that avoids the limitations of NMR with paramagnetism, and it will provide some precise results and description of our molecule's geometry in its crystalline form. The problem is we need to actually obtain our products in their pure crystalline form, which can be difficult to get them pure, and to have them crystallize can take multiple attempts, but that is an ongoing process. Some we have attempted, and some we hope to in the future to obtain crystals and get X-ray crystallography analysis from them. Also, we can react with more metal reactants, try a wider range of metals, and also combinations of two different metals to react with the ligand. Here are some items for further research, and some of these I mentioned already. These new compounds can have further characterization. New metals or new combinations of metals can be experimented with. Their catalytic properties can be assessed, and if there are some small molecules that are activated by these metallated ligands, then some practical applications can also be explored. But even in the lack of practical applications, this research can provide a contribution to the greater field of amphiphilic ligands.